Hello dear students, welcome to Biology Decrypted. In this series of Biochemistry Decrypted, we have covered lecture 1, that is Chemistry Fundamentals Part 1 so far. Now in this particular session, we will be talking about Chemistry Fundamentals Part 2, and we will be discussing an important topic today, that is chemical bonding and interactions. So we'll be talking about chemical bonding and interactions. Now, to start with, we'll be talking about bonds. Well, bonds can be formed in different ways. There are a few ways to form bonds. First, we'll be talking about the method of sharing of electrons. We had already discussed about electrovalent bonds and covalent bonds in the previous session. So if you haven't watched the session yet, do watch it and then continue with this session. So, firstly, bonds are formed by sharing of electrons. So what happens here is, obviously there will be an orbital with a single electron, unpaired electron. Similarly, there will be another orbital, and they can either undergo head-on overlap like this. So this is an example of head-on overlap. And this is a strong overlap, and due to this strong overlap, the bond which is formed is a sigma bond. This is called sigma bond. Fine. So this is a sigma bond. Obviously, it is a covalent bond. So because of sharing of electrons, the bonds which are formed are called as covalent bonds. As you can see, the name itself suggests covalent. So what about the pi bonds? We had discussed about pi bonds as well. For example, if carbon has these hybrid orbitals, for example, in this way, it has various hybrid orbitals. Let's say this is sp3 hybrid orbitals. These are sp3 hybrid orbitals. Now, what can happen is they can either form bonds with other sp3 hybrid orbitals, or they can form bonds with pure s or pure p orbitals. The only condition being they will be forming these bonds by head-on overlap, and which would obviously lead to the formation of sigma bonds. Now, there is also a possibility in which a few orbitals stay unhybridized. Like p orbital stays unhybridized, and these unhybridized p orbitals, which are present in a perpendicular fashion, they will actually end up doing this kind of a sideways overlap. They won't be colliding heads on, rather they would be actually forming a bond in a sideways fashion. So this is an example of pi bond. What happens is unhybridized p orbitals, they undergo sideways overlap, and therefore they form pi bonds. So the bonds appear like this. So this is the way in which pi bonds are formed. This is an example of pi bond. So this is how basically by sharing of electrons covalent bonds are formed. Now, bonds can also be formed via electron transfer. So electron transfer can happen, and as you know, electron transfer in case of sodium, Na loses an electron to give you Na plus. Similarly, chlorine, it gains an electron to give you Cl minus. Now you have two ions, that is Na plus and Cl minus. So the first one is the cation and the second one is an anion, and between these two charged species, between these two charges, that is Na plus and Cl minus, between these ions, there are interactions that get formed. This is actually called an electrovalent bond. This is called as electrovalent bond or ionic bond. So what happens here is actually the electrons are transferred and they are not shared. There is a difference between sharing of electrons and transfer of electrons. Transfer of electrons will lead to the formation of charged species. Some will have the electrons, some will lose the electrons. So the ones that lose the electrons are electropositive, the ones that gain electrons are electronegative, and the species that eventually form are charged species called ions. And if we talk about sharing of electrons, what happens is I am investing one electron, you are investing another electron, and this is how both the electrons are being shared in this manner. So there is a difference between covalent bonds and electrovalent bonds. So these are the two main methods. There is another method. There is another method of formation of bonds. This method actually involves both. There is sharing as well as transfer happening. So sharing and transfer, both the events take place. So what happens here is a dative bond, which is also known as a coordinate covalent bond is formed. So what happens when sharing and transfer, both the phenomenon take place simultaneously? So what happens here is, for example, you can take an example of ammonia. It has a lone pair. This lone pair is given to someone who needs it, like H plus. So this lone pair is given to H plus, and this is how a dative bond or a coordinate covalent bond is actually formed. So now we know that there are three major methods of formation of chemical bonds, the strong bonds especially. So covalent bonds, electrovalent bonds, as well as dative bond, or you can also call it as coordinate covalent bond. So see, it is a coordinate bond because it has a direction, but also it is covalent because if you see, two electrons are being shared, that is, the lone pair is being shared. This one has both the electrons, this is empty, but when this sharing is being done, does it matter that from where the electrons came from? Yes, we do signify it with an arrow that it came from this particular atom, but when it is shared, it doesn't matter because it is being shared after all. So this is the thing. We just show it with an arrow, but the overall concept here is it is also a covalent bond. So therefore it is called as a coordinate covalent bond. Now, moving forward, are these the only bonds that are formed in biological systems? Well, no. There are some other forces involved as well. These forces are called as intermolecular forces. And these are also called as molecular interactions. So, if you talk about these intermolecular forces or molecular interactions, basically what happens is these are not formed within the molecule. Rather, what happens is between two different molecules, these interactions exist. So, if you talk about hydrogen and hydrogen, this is an example of hydrogen molecule. Rather, let's talk about hydrogen fluoride, HF. There is another molecule of HF, and between HF and HF, there will be some kind of intermolecular force involved. Just like in case of water. If you take the example of H2O, so between the molecules of water, there are hydrogen bonds that are involved. Even here, there are hydrogen bonds involved. So these are examples of intermolecular forces, or you can say molecular interactions. These are examples of molecular interactions. Just a moment. Yeah. So this is an example of intermolecular forces between water molecules. So these are all examples of molecular interactions. Now, let's talk more about bonds. Bonds can be strong or weak. So there can be weak bonds and there can be strong bonds as well. So weak bonds, strong bonds, both type of bonds exist. These weak bonds, they have a tendency to get broken very easily. So weak bonds break easily, and strong bonds, they do not break easily. Now, what happens is if we try to assign a certain amount of energy, the energy requirement to break these bonds. I hope you remember the fact that we discussed in the previous class that bond breaking involves requirement of energy, whereas bond formation involves release of energy. This is very important. Keep this in mind forever. So, bond energies. We'll talk about bond energies, or we can say bond enthalpies. The unit that is assigned here is kilojoule per mole. This is the unit in which we measure bond energies, and we'll assign the values of approximate values of bond energies to both the bonds. For weak bonds, if you talk about weak bonds, it is less than 40 kilojoule per mole of energy. Whereas if you talk about strong bonds, it is in the range of about, you can say it can be somewhat more than 400 kilojoule per mole. So this gives you an idea how weak these weak bonds are and how strong these strong bonds are. But remember one thing, these weak bonds can outnumber the strong bonds as well. How? Well, by increasing the number. What happens is, if these weak bonds, if they increase their number, for example, if the number of weak interactions, goes up, what can happen is the overall strength, overall magnitude will get increased. So the overall magnitude will also increase if the number of weak interactions increase. So if there are many weak interactions happening over a large surface area, this can result in a greater value as compared to a single strong covalent bond. So yes, these weak bonds are found in biology in abundance, and these are responsible for many biomolecules, like many biomolecular structures. Like this is responsible for secondary as well as tertiary structures and stability of various biomolecules. As you might be knowing, there are these globular structures of proteins which are found, and in these globular structures of proteins, what is found is in these tertiary structures, actually tertiary structure of proteins. Also in case of DNA, in case of RNA, you do find various weak interactions like hydrogen bonds, hydrophobic interactions. You also find van der Waals forces. So all these kind of interactions are found in various biomolecules across biology. So this is the basic concept of strength of bonds. That is, there are weak bonds as well as there are strong bonds. Now, moving ahead, let's compare various bonds. So first of all, the way of classifying them is bonds can be classified as covalent. So we can include interactions as well. Not all bonds are interactions. Remember it that not all interactions lead to the formation of bonds, whereas all bonds can be considered as interactions. So if we try to classify bonds or interactions, so there can be covalent interactions or covalent bonds, and there can be non-covalent interactions. In biological systems, if we talk about covalent bonds include many bonds, like there are peptide bonds. Peptide bonds can be represented in the form of CONH. Then there are ester bonds, C double bond O, O R dash. These are ester bonds formed between acids and alcohols. Then there are anhydride bonds, which are formed between carboxylic acids, C double bond O, O, C double bond O, like this. Then there can also be disulfide bonds. Disulfide bonds can be formed in this manner. Then there can be ether bonds, ether linkages, or you can call these as glycosidic bonds. Single bond, O single bond, in this way. So there are many types of covalent bonds that can actually form. There can be another type of bond in which there is a mixed anhydride bond between a phosphate and a carboxylic acid. Then there can be phosphodiester bonds. So there are various types of covalent bonds that are possible in biological systems. Now, talking about the non-covalent interactions, there can be some interactions in which covalent bonds are not being formed. So non-covalent interactions include ionic bonds, or these are also called as salt bridges in biological systems. Remember, ionic bonds in vacuum are the strongest, but when you compare compare the strength of ionic bonds as well as covalent bonds in biological systems, that is in aqueous systems, what happens is due to the reason that water is a polar solvent and ionic bonds involve charges, what happens is solvation takes place, and due to this solvation, due to these multiple interactions being formed between water molecules as well as the ions, what happens is the bonds actually get weakened. So that's why in aqueous solution, aqueous medium, ionic bonds are not as strong as they are in vacuum. So yes, the magnitude of ionic bonds in case of biological systems is lesser. So in biological systems, covalent bonds are the strongest, because yes, covalent bonds, generally the non-polar covalent bonds, they do not interact with water. So water doesn't lead to an effective solvation of these bonds, and that's why these remain the strongest in aqueous systems. Now, ionic bonds or salt bridges can be example of non-covalent interactions. Also, hydrogen bonds. Hydrogen bonds are a good example of non-covalent interactions. Also, there are hydrophobic interactions. And also there are van der Waals interactions. Now, let us look at the amount of bond energy that is required to break each of these bonds. Now, to start with, let's start with covalent bonds. For covalent bonds, the value of energy that is required is approximately in the range of 250 to 450 kilojoule per mole. Now, if we talk about ionic bonds in biological systems, of course, ionic bonds, or these ionic bonds are also called as ionic interactions. They can also be called as salt bridges. Basically, these can form between any of the charges, like if for example there is an NH3 plus group, and for example if there is O minus, so there can be ionic interactions that get formed between these particular charges, So these are example of ionic interactions or salt bridges, which are found. These can be found in these can be found in the surface of various proteins. These can be found in other biomolecules like DNA, etc. Now, moving forward, what is the approximate value of energy for these particular bonds? So this is a range. Obviously, there is a lower limit, there is an upper limit. So we can say that approximately between 6 to 40 kilojoule per mole of energy is required. And as I told you that these ionic bonds, they are not generally weak, because in chemistry we have already learned that ionic bonds are actually very strong. But what happens is, in biological systems, they get weak only in presence of water, that is in aqueous solutions, or you can say aqueous systems. And this happens because of the phenomenon of solvation. I hope that's clear now. Now let's move forward. Let's talk about hydrogen bonds. Now, if we talk about hydrogen bonds, what happens is, hydrogen bonds, to break these hydrogen bonds, we require 12 to 30 kilojoule per mole of energy. And this is how we can say that it is weak, and weaker than ionic bonds, of course. And the major contributing factor behind these hydrogen bonds, as we'll see later on, is also electrostatic. So the basic fundamental concept behind this is electrostatic. So the basic nature is electrostatic. Also, similarly, if we talk about hydrogen bonds, in case of hydrogen bonds also, the nature of interactions is electrostatic. Fine. So these are electrostatic as well. Now talking about a few other interactions, that is, we'll be discussing about the hydrophobic interactions. These hydrophobic interactions, they have a bond energy requirement in the range of 4 to 12 kilojoule per mole. And if we talk about van der Waals interactions, their value lies in the range of 2 to 4 kilojoule per mole. As you can see that these are the weakest interactions, but as I told you, if there are many such interactions together, they can easily outnumber the strength of covalent bonds as well. Now, moving forward, let's talk about covalent bonds in detail. covalent bonds. These covalent bonds, they have, so what happens is, basically we can denote these covalent bonds in terms of equal sharing of electrons. So what happens is, we can take this example of A and B, where A and B are two atoms that are involved in a covalent bond. And now we will be drawing the electrons like this. This is how we have drawn A and B. Now they are sharing the electrons. A requires one electron to complete its octet. Similarly, B also requires one electron to complete its octet. And this is how they share these two electrons, and this forms a covalent bond. This can be represented in the form A bond B. And here we can see that there is equal sharing of electrons. Equal sharing is taking place. Now, had it been a coordinate covalent bond, had this been a coordinate covalent bond instead of a pure covalent bond, so coordinate covalent bond. In this case, actually, A would have had a complete octet and B would have been electron deficient, and here A would donate this lone pair to B, and it would be donating as well as there would be sharing involved. So we would call it a coordinate covalent bond. So what happens is, this will be donating this to B, and when we see, if we try to show it in the form of sharing, this would be the story. So we would represent this as A to B. Also, let me tell you one more thing. The one in a coordinate bond, the member in a coordinate bond which donates electrons, which actually gives electrons or lone pair, is called as a Lewis base. And the one which accepts is called Lewis acid. This is the basic concept. Now, covalent bonds have a unique tendency to get polarized. So what happens is, these bonds can get polarized. And what is the source of this polarity or polarization? Basically, what happens here is, there is a concept of electronegativity. Now, this concept of electronegativity, or EN, talks about a bond, a covalent bond, in which there are two atoms, A and B, and here one atom, let's say B, has a greater electronegativity as compared to A. For example, you are atom A and I am atom B. So we are having a shared pure covalent bond, but I have a tendency to attract a shared pair of electrons towards myself. For example, we have shared one electron each, but what I am doing is, I am trying to keep both the electrons near myself. And what is happening in this case is, the electronegativity, like my electronegativity is greater and your electronegativity is lesser, and the bond which was initially thought to be covalent is now getting polarized. So what is happening here is, polarity is slowly developing. I am becoming delta negative, slightly partially negative, and you are becoming partially positive. So this is how a bond got polarized. This thing itself is called polarity, and we can say that polarity is being induced, and therefore the bond is said to be polarized. Now we can see here that one member has greater electronegativity, another has lesser electronegativity. So what we can say here is, there is an electronegativity difference. There is delta En, difference in electronegativity. So we can call this as electronegativity difference. Now, we can take some examples. For example, if there is bond between ClCl, if there is bond between OO, so here the atoms are of the same type. There are same atoms, and same atoms would have same electronegativity. And since they have same electronegativity, what we would see here is their delta En, their value of delta En would be 0, because they won't have any kind of electronegativity difference. The electronegativity difference would be 0, and these type of bonds are pure covalent bonds. There is hardly any kind of polarity in between them because there are same atoms. So the electronegativity difference would be 0. But what if there are different atoms? What if there are atoms between which there is a huge electronegativity difference? In that case, the bond will be polarized to a greater magnitude. For example, if there is a bond between C and N, if there is a bond between C and O, for example, if there is a bond between H and F, all these bonds, all these covalent bonds are not purely covalent, as all these bonds are polarized in this way. So all these are examples of polar covalent bonds. All these are examples of polar covalent bonds in which there is a significant electronegativity difference. And this is how the bonds are polarized. So we saw this important concept of electronegativity and bond polarity. Now let's take examples of covalent bonds. So here, as you can see on this slide, there are a few values of covalent bonds. Now let's talk about this. What you can see here is the respective bonds and their corresponding bond energy in the units kilojoule per mole is given here. Now talking about HH, for example, hydrogen. In case of hydrogen, if you see the bond energy is 436 kilojoule per mole. For CH, it is 414 kilojoule per mole. For CC, it is 343 kilojoule per mole. For Cn, it is 292 kilojoule per mole. For CO, it is 351 kilojoule per mole. For C double bond C, that is in alkenes, it is 615 kilojoule per mole. For C double bond N, it is 615 kilojoule per mole. For C double bond O, it is 686 kilojoule per mole. For O single bond O, it is 142 kilojoule per mole. For O double bond O, it is 402 kilojoule per mole. For N triple bond N, nitrogen gas, the value is tremendously high. It is 946 kilojoule per mole, and therefore it doesn't break easily at room temperature, and it behaves in a way that as if it is chemically inert. Now NH bond, it has a bond energy of 393 kilojoule per mole. OH bond has a bond energy of 460 kilojoule per mole. So these are a few values of bond energy of covalent bonds. Now let me tell you about an important concept. An important concept here is about bond order. Now, if we talk about bond order basically signifies the number of bonds. So what happens is, if the number of bonds increases, individual bonds will become shorter in length, and due to this we can say that it is inversely proportional. Bond order is inversely proportional to bond length. Okay, so let's write this again, that bond order is inversely proportional to bond length. Also, what we can say here is bond order, the number of bonds, greater the number of bonds, the difficult it is to break it. So we can say here that bond order is directly proportional to bond strength as well. Bond strength, or we can call it as bond energy. So bond order is directly proportional to bond strength, or we can say bond energy. So yes, it is bond strength. And as we know, all this is inversely proportional to bond length. So this is the overall concept of bond order, bond energy, or we can say bond strength, and bond length. So these are the relations what we can see here. On the basis of bond energy, we can also comment on bond length, we can also comment on bond strength, bond order as well. Now, moving ahead, let's move ahead. Let's talk about the electrostatic interactions or ionic interactions or the salt bridges. We have talked enough about covalent bonds. Now let's come to electrostatic interactions. Electrostatic interactions or they are also called as ionic interactions, or we can also call these as salt bridges. These are the interactions. Now, taking example of Na plus Cl minus, there will be strong electrostatic forces of attraction here, and this force can be given by a formula. This force, which is also called as Coulomb's force of attraction, we can say F is equal to 1 by 4 pi epsilon, 1 by 4 pi epsilon into Q1 Q2 upon R square, where this epsilon term, as I had already told you earlier, is dielectric constant. This is called as dielectric constant, and it is specific for a medium. Especially we can talk about solvents as medium. Also, this is called as relative permittivity. This is also called as relative permittivity, and it is generally measured at 25 degrees Celsius, which is the room temperature. So now, moving ahead. If we see here, F is inversely proportional to R square. F is inversely proportional to R square. So what happens is, if we plot F and R square, we'll see that there it follows inverse square law. If we take a low value of R, we see that the force is very high. Now when this separation is being increased, the force is decreasing and gradually decreasing. So this is the graph between F and R square. Now, if we do not consider a particular medium, if we, let's say, take an example of vacuum. In case of vacuum, this epsilon is called as epsilon naught, or it is also called as the absolute permittivity. It is also called as absolute permittivity, or it can be also called as permittivity of free space. Permittivity of free space. Now, if we talk about an important medium or solvent in biological systems, obviously we'll talk about water. In case of water, if we try to find out the epsilon value for water, or the value of dielectric constant for water, is approximately 78.5. We consider it roughly as in some cases roughly we consider it as 80 for water. So but yes, the actual value is somewhere around 78.5 to 79, but yes, 80 is also considered in some places. So what we see here is that actually water has a very high value of dielectric constant. So we can say that it has a high value of dielectric constant. And this is actually a major factor why, a major reason why water is considered as a universal solvent. This is one of the reasons, one of the major reasons. Also, there is another reason. There is another reason of dipole moment. So let's talk about dipoles and dipole moment. Now, talking about dipoles, water is an example of dipole. HOH. What we can see here is that the bonds are polarized here. Delta positive, delta negative, delta positive. Now, what we can see here is that this is acting as a dipole. How do we represent dipoles? We represent dipoles in this format, where this is the positive part, this is the negative part. This can be partial positive, partial negative as well. It can be completely positive, completely negative. But yes, from positive to negative, we show this arrow, and this arrow actually represents dipole moment. This represents dipole moment, and dipole moment is mathematically represented as mu. So the symbol for dipole moment, mu, is the symbol for dipole moment. And one more thing, the unit of dipole moment is d, capital D, or we can also call it as dBi. And the formula for mu is E into D, where d is the distance or the dipole length, and E is basically the magnitude of charge which is present. And since the charges are equal in magnitude, just opposite in direction, so we just take the modulus value of the particular charges. So this is the actual concept behind it. Now, this is one dipole and this is another dipole. Now, if we extrapolate it here, we can write it in this way, and this will have a net resultant dipole moment in this direction. This will be the net resultant, just like vectors. This has a resultant dipole moment. Now, as we can see, water is polar. Water has a net dipole moment. What about carbon dioxide? Does carbon dioxide have dipoles in its structure? Let's see. This is the structure of carbon dioxide. Now, in case of carbon dioxide, what we can see here is this is delta positive, this is delta negative, this is delta negative. We can see that it has dipoles. It has dipoles in its structure, and the dipole moment will be in this direction and in this direction respectively. But we see an important thing here, that the angle between these two is 180 degree, and when these are present in exact opposite directions, what we can comment here is it will cancel out each other. So the magnitude is same for both the dipoles and the direction is exactly opposite, that is 180 degree. What will happen here is they will cancel out each other and the net dipole moment will be 0. Whereas in case of water, the dipole moment will be non-zero. It will be non-zero. It will have a certain specific value. So this is the concept of dipoles and dipole moment. Now let's move ahead. Now we'll be talking about hydrogen bonds. Very important. Hydrogen bonds. Talking about hydrogen bonds, what we see here is generally there is a donor, there is a hydrogen bond donor always. And this is the atom to which hydrogen is attached. So hydrogen is attached covalently to this particular atom. This is the donor. And generally what we see is that this donor has an appreciable amount of electronegativity. So it is actually, electronegativity is high, obviously higher than hydrogen, and therefore it will already polarize the bond. This will have delta negative, this will have delta positive. Now, there will be an acceptor as well. So what we see here is there is an acceptor as well, hydrogen bond acceptor. And this acceptor is also having electronegativity. So this is also electronegative. And it actually has delta negative charge because it is electronegative. It has actually it has pulled the electron clouds towards itself, and it is having an appreciable amount of electron density, and therefore it is delta negative. Now what happens here is, due to this charge and this charge, there is an electrostatic interaction being formed, and this electrostatic interaction between the acceptor and the hydrogen, and also we can comment on this hydrogen that this hydrogen is an acidic hydrogen. What does this acidic hydrogen mean? An acidic hydrogen is a hydrogen that can be easily lost in the form of H plus. So what would happen here is, this donor would actually pull these electrons, and H plus will be lost. H will be lost in the form of H plus. So this is basically an acidic hydrogen. Acidic hydrogen. So these acidic hydrogens are the targets of hydrogen bonds. What happens here is, this acceptor comes, and this acceptor, which has a delta negative charge, forms an electrostatic interaction, which is actually called a hydrogen bond. So this is called as the H bond or hydrogen bond. And what we can say is, these particular atoms, the donor and acceptor pairs, are generally fixed in biological systems as well as in the whole of chemistry. We can say that electronegativity is the highest for these particular atoms, that is F, O and N, fluorine, oxygen, nitrogen. And if we talk about the order, fluorine has the highest electronegativity, then oxygen, and then comes nitrogen. So what happens here is, in these hydrogen bonds, we can see that these hydrogen bonds are formed between these particular atoms, fluorine, oxygen, nitrogen, or we can say phone. So yes, fluorine, this hydrogen bond is formed between oxygen and hydrogen, which is attached to fluorine. Also another case is possible, that H is attached to N, and then this forms a hydrogen bond with oxygen. And in a similar way, many such cases are possible. If we take an example of water, HOH, delta positive, delta negative, then what happens here is, another water molecule comes here, and this is how hydrogen bonds are formed. And we can see that hydrogen bonds are found in abundance in case of water as well, and this is the reason why water is liquid in nature. Now, hydrogen bonds. These bonds can be either intramolecular, or hydrogen bonds can be intermolecular. What do these terms mean? Intermolecular means between two different molecules. This simply means between two different molecules. What we can see here in case of water, we can see that it is formed between two or more different molecules. Whereas intramolecular is between the same molecule. If hydrogen bonds are formed within the same molecule, then these hydrogen bonds are called as intramolecular. Whereas if these are formed between two different molecules, then this will be called as intermolecular hydrogen bonds. Now, there will be certain fixed criteria for hydrogen bonding. Talking about hydrogen bonding criteria. Criteria for hydrogen bonding. If we talk about the criteria, one important point is the electronegativity difference between the donor and acceptor. There should be electronegativity difference between donor and acceptor, and only then this arrangement will be possible, where this is delta negative, this is delta positive, and this is also delta negative, and this hydrogen bond will be formed. Also, there is another important point, the alignment or you can say the orientation. The alignment or orientation of the atoms is also very crucial for hydrogen bond formation. Let's take two examples. For example, if in this case there is donor, hydrogen, and the acceptor, this is how a hydrogen bond is formed, and you can see this is a perfect straight alignment. This is a perfectly straight alignment, and the angle is 180 degree here. That is, they are exactly opposite. They are exactly linearly present. So here what we can comment is the strength will be maximum in this case. Whereas if there is another scenario, if the donor is present here and the acceptor comes at a different angle, let's say this angle is somewhat 130 degree. Now what we see here is the strength will be obviously lesser than the first case. This is the first case and this is the second case. What we see here is that the strength in second case is strength of the hydrogen bond in second case is much lesser as compared to the first case, because the alignment is not in this manner. This is instead in this manner, and this is not how an effective electrostatic interaction is formed. Therefore, this orientation or alignment affects the strength of hydrogen bonding. Now, let's come to another important interaction, that is intermolecular interactions or forces. So let's talk about intermolecular interactions or forces. So if we talk about intermolecular interactions or forces, we will be talking about van der Waals interactions. And van der Waals interactions, whenever we talk about these interactions, these are common to non-polar as well as polar compounds, common for all. And van der Waals interactions is basically a broad group in which we classify it into various other type of interactions, and we'll be classifying these one by one. First case, this is about dipole-dipole interactions. Dipole-dipole interactions. Now, important to note that these are permanent dipoles. So permanent dipole interaction. These forces are also called as Keesom forces. And what happens here is both are permanent. So both are permanent dipoles. Let's take example of these dipoles. First case is delta positive, delta negative. The other one is delta positive, delta negative. Similarly, if we take another example here. So what happens here is simply there is electrostatic interaction here because these were dipoles from the very beginning. Just like there are permanent magnets. So two permanent magnets interacting together. In a similar way, two permanent dipoles interacting together, these lead to Keesom forces. These are also called as Keesom forces, the forces between these kind of dipoles, permanent dipoles. These come under van der Waals interactions. Now another one, let's talk about the other one. We'll be talking about Debye interactions. Debye interactions. If we talk about Debye interactions, what we see here is that there are permanent dipoles. There is one permanent dipole, and the other member, the other participant here is an induced dipole. What is an induced dipole? Initially, it was not a dipole. But what happened is when it came in contact with a permanent dipole, it actually dipoles were introduced. So it was introduced, or you can say it was induced by this permanent dipole. So while we were keeping this simple thing in front of this molecule in front of this permanent dipole, this already had this was already a dipole, but in its influence, what happened? This induced dipole developed, and this developed partial positive and partial negative charge, and therefore it was called as a induced dipole. So this is how an induced dipole was formed, and this led to the formation of Debye interactions. Now, moving forward, if there are if for example there are two molecules in which none of them is a dipole, but what happened is one instantaneously it developed a dipole. What happened is electrons, electrons were moving from throughout the molecule, and they started accumulating in a particular region. Due to this, there was partial negative charge developing in this part. This developed partial negative charge. Similarly, the other part developed partial positive charge, and this is how an instantaneous dipole was formed. So instantaneous dipole was formed, and due to this what happens? So dispersion of electrons was taking place, and therefore these are also called London dispersion forces. So this is called as London dispersion forces. So moving ahead, because of this, delta negative and delta positive was formed here, and this was an induced dipole which was formed. This was an induced dipole. So basically what we can call it is as induced dipole. So this is an example of induced dipole. Similarly, this is an example of permanent dipole, induced dipole. This is an example of permanent dipole. So these are various different types of interactions which are classified under the heading of van der Waals interactions. So this can form between non-polar as well as polar molecules. And now let's plot it graphically. Let's plot the intermolecular potential energy, that is IMPE, with respect to the separation or distance between the molecules. Let's draw a graph. So this is how we draw the axis. This represents positive, this represents negative, and this whole thing, this represents the separation, or we can say the distance, separation or distance. Now here on the y-axis we have intermolecular potential energy, or we can call it as IMPE. Now what we see here is actually when the separation is much larger, there is hardly any amount of potential energy. Then gradually when we reduce the distance, what we see here is that there is a minima at which there is maximum amount of bond formation, but beyond that, if you bring it closer, what will happen is it will increase and increase, and it will increase infinitely. So this is the graph which we see here, and what we can say is that this is epsilon, and this is a measure of strength of attraction, and this is the region called sigma, in which the IMP potential is 0. And we can say that this intermolecular potential energy, or this energy, can be represented by two major parameters: a parameter for repulsion and a parameter for attraction. So for attraction, the value is, obviously there will be a negative sign for attraction, B by r to the power 6. And if we talk about repulsion, it will be positive, and A by r to the power 12. This is how we represent repulsion component as well as attraction component as this van der Waals force. If you try to do this experiment, if you try to keep these fingers just very close to each other, but you are not applying any kind of force and just observing it, what you can see here is that the interactions are forming and breaking. This is how you can visualize van der Waals forces in real life. Also, van der Waals forces can be seen in case of these you must have heard about gecko, lizards. So these have in their digits, they have fine bristle like projections, and these form van der Waal interactions with the wall surfaces. So talking about repulsion, it is inversely proportional to the power 6. And similarly, attraction, if you talk about the attraction component, it is for repulsion it is 10 to the power, it is r to the power 12, and attraction is inversely proportional to r to the power 6. So this is the overall concept of repulsion attraction in case of van der Waals forces. And if you try to visualize, what happens is there are two molecules. For example, this is one molecule and there is another molecule in this way. This is another molecule. This is another molecule. So what we can say here is that this, in this range, this is the separation. In this separation, in this range, actually van der Waals interactions will take place. So this is the overall concept of van der Waals interactions. Now let's talk about one last interaction, that is the hydrophobic interactions, which is also called as the hydrophobic effect. So these hydrophobic interactions, this effect which takes place is also called as hydrophobic effect. Now, talking about hydrophobic interactions or the hydrophobic effect at large, what we can say here, we can take an example. For example, there is bulk water here and we are dropping oil, drop by drop, drop by drop. So what happens here is that you have generally seen this, that oil, these oil droplets will eventually disperse, but after some time they will form a layer and they would separate. So if we talk about these non-polar substances, these will actually form a layer over water, which is a polar solvent. This is the non-polar substance, whatever we disperse in a polar solvent like water. So what happens is they have an inherent tendency to aggregate, and this aggregation is occurring. We can actually sense a force, some kind of a force which is actually bringing them together, especially when they are surrounded by water, because they won't interact with water, because obviously like dissolves like and water won't dissolve oil. So what would happen is they would have a common hatred for water, and guided by this particular force, guided by this particular hydrophobic interaction, this force, they would be actually coming together and they would be interacting. What would happen is, initially when these oil droplets would be dispersed, they would be surrounded by water molecules, and water molecules will have this unusual tendency of forming cage-like structures. Water molecules, through their unique property of hydrogen bonding, they would form cage-like structures. And now these cage-like structures are actually ordered with respect to the other water molecules in the bulk. These will be more ordered, and since these will be more ordered, we can say that the overall entropy change of this process will be negative, because this is the final state, this is more ordered. Since the final state is more ordered, the entropy change will be negative. And if we actually put this in the equation, delta G is equal to delta H minus T delta S, what we would see here is delta S is negative, due to which this whole term will be positive, and delta G would eventually result in a positive value, making these interactions unfavorable. If there are individual cage-like structures like this, multiple cage-like structures like this, this would actually become unfavorable. So there is a way. If the number of cage-like structures is reduced, what happens is, if these particular molecules, they come together, if they aggregate together, and there is one cage instead of multiple cages, if there is one large cage instead of multiple cages, what would happen here is that the water molecules will be free from these hydrophobic surfaces, and they would finally form lesser number of cages, and due to which actually this will be the most favorable interaction, and this is the guiding force. The guiding force here is the interaction between these particular molecules. See, there is no such specific interaction, but yes, because everything is being done to avoid the contact of water, that's why they are coming together and hydrophobic effect is taking place and these hydrophobic interactions are formed. So this is all for chemistry fundamentals part 2. We have discussed many interactions, chemical bonds as well as interactions. In the upcoming lecture, chemistry fundamentals part 3, we will be talking about an important concept of solutions and colligative properties. So this will further strengthen your fundamentals of chemistry, and we will be starting mainstream biochemistry a few lectures later. So this is all from Biochemistry Decrypted lecture 2. Thank you so much for watching. Do share this video with your friends. Do let them know about this free lecture series. Do like this video and also give your valuable comments so that we can make this even better for you. Thank you so much for attending. See you all in the next one.